It's still killing over 500,000 children under the age of 5 in sub-Saharan Africa and Southeast Asia every year. The vaccine that you may have heard about is less effective than a bed net. The drug that was developed about 15 years ago, it's called artemisinin. Those compounds are now no longer killing parasites effectively in much of Southeast Asia, and those resistant parasites are now in sub-Saharan Africa. So we really desperately need new, highly effective solutions, both vaccines that prevent infection and drugs that cure infected individuals. We identified a vaccine candidate that's on the parasite. It's a parasite protein. The parasite puts it on the surface of the infected red cell. And when that protein gets bound by antibody, the parasite commits suicide, it dies. That antibody binding alone kills the parasite. And so just recognition transmits a kill signal. If antibody binding kills the parasite, maybe we could get a small molecule drug that would mimic the binding of an antibody, that would allow GARP to get activated, that's our vaccine protein, and kill the parasite. And so we did a screen looking for small molecule drugs that would mimic the binding of antibody. And we screened thousands and thousands and thousands. The target and the compound we have would be ideal therapies for severe malaria. And I would personally like to see this compound reserved for severe malaria so that we actually start implementing classic antibiotic stewardship for malaria. So our vaccine has three components. It's got a component that generates antibodies against GARP, and so targeting the exact same pathway as the drug. It's got a component that generates antibodies against Schizont egress antigen. That traps the parasite inside the red cell. And it has a component directed against another protein which prevents the parasite from entering the red cell. So entry, intracellular development, and egress is the vaccine, whereas the drug is just intracellular development. In a nutshell, I think the parasite puts GARP on the surface to try and limit parasite growth. Parasite doesn't want to kill you. Remember, when it kills you, it's a tragedy for the person. It's also a tragedy for the parasite because the parasite will never jump to the next host. So the best parasites are parasites that grow to the maximum density possible inside you, but no further. They want to grow until you're sick, but not grow until you're dead. And so we think GARP is critically involved in that process, and we're using that same pathway to drive parasitemia to zero. We've demonstrated that the drug binds to GARP using a variety of biochemical approaches. We have demonstrated that the drug kills parasites in tissue culture. So we grow parasites in the lab, add this drug, parasites all die. We've demonstrated that we can deliver the drug to animals and achieve blood level concentrations that should be lethal to the parasite. We've demonstrated that it's safe for the animals. We've demonstrated that there's no measurable toxicity to human cells. And we've taken this into an animal model where we infect the animal with real-life human falciparum malaria, and we treat the animal with our compound, and that compound kills the parasites in the animal model. So I know that it doesn't hurt the growth of human cells in culture. I know it doesn't hurt human white blood cells, and I know it doesn't do anything given at high doses to mice. So I'm pretty comfortable. Now, there's more work to be done, don't get me wrong, but we have not seen a safety signal, as it's called, given the testing that we've done so far. That vaccine is ready for human development. And what I mean by that is it's on the same lipid encapsulated mRNA platform as the BioNTech Pfizer COVID vaccine. So the platform, the delivery platform, has already been, extensively scrutinized by the FDA. So we know what the roadmap is for moving into human trials. What it takes is the capital resources to do the manufacturing of the vaccine under conditions that are appropriate for use in humans. We need a very limited toxicity panel because the platform has been so well established. So it's a limited toxicity platform, and then we're doing a phase 1 study. That easily can be done in 18 months.